Okay, so of course, lab centrifuges are not like the little model that you saw in the previous slide. They're much more complicated than that. And I gotta say, from working with centrifuges a lot, they're one of the most dangerous things in the lab. They go so fast, and there's so much force that's actually imposed by these machines that you really want to be careful around them. So, what you can see here what you can see here is a researcher putting the sample into, it kind of looks like a washing machine almost, right? It's, this is the centrifuge, and it's computer controlled, and it spins around to the point where, the dense stuff starts to separate. So you start it generally in stages where you would go, 1,000 Gs, right? So that's 1,000 times the force of gravity for 10 minutes. Then you take out the stuff at the top and put it in the next tube, and that goes to 20,000 Gs for 20 minutes, right? And you keep going until you're up to 150,000 Gs for 3 hours. And what that does is eventually pellets, or in other words, gets all these little dense parts of the cell to sink. And by doing these in successive steps, you can separate, different aspects of the cellular material, right? So I just want you to realize what, the forces here. 150,000 Gs. Right now, I don't know about you guys, but I'll share. I weigh about 210 pounds, right? And that's at 1 G, right? So I feel like I weigh 210 pounds right now. And that's what my body's used to. At 150,000 Gs, multiply my 210 pounds by 150,000 times the force of gravity, I would literally be mush, right? I mean, I would not be even able to hold myself up. My bone structure would not be able to hold myself up under those forces. So really, really, really crazy amounts of force here, but that's what's needed to separate all these little pieces of and components of cells from each other for study. So obviously, that's an important thing to consider. So let's talk about the difference between eukaryotic and prokaryotic cells. Okay, so eukaryotic cells are really what most of the cellular life in the world is composed of. Okay, so as far as organisms that are you know, uh, multicellular, I guess is probably the best way to say it. So, in other words, large complex organisms, right? Um, so that means that there are obviously other cells that are not that complex, and they don't form multicellular organisms. So that's what we're going to be doing in these next couple slides is we're going to be comparing the two. So the basic structural and functional unit of every organism is one of two types of cells. Prokaryotic, which are the simplest ones, and eukaryotic, which are the more complex ones. So the only organisms that are prokaryotic, okay, so pro basically means before, and then karyotic means nucleus, okay, so would be bacteria and another sort of kind of form of bacteria, which we call archaea, okay. Archaea and bacteria are actually distinct enough where they can be separated into two different kingdoms, but they are still prokaryotic, okay, so in other words, they do not have a nucleus. They were probably on the Earth, or at least similar organisms to this were probably on the Earth before a nucleus evolved in, cells that we call eukaryotic, right? So eukaryotic cells, this means true nucleus, okay, um, consists of protists. These are the little things that swim around in pond water. There are some parasitic forms of protists, fungi, okay, animals, and plants. So these are much more familiar organisms to us, I think, on the whole. But obviously, bacteria and archaea are really important to consider too. They have a huge effect on living things on the planet. So we'll be talking about those very briefly, and then we'll get into more cell components. So the basic features of all cells. Now you can already know, by just looking at this first statement, wow, that makes a really good quiz question. Like, what's the difference and what's the similarities between the two types of cells we just mentioned, prokaryotic and eukaryotic, right? Well, all cells have a plasma membrane. You can also call this a cell membrane. Um, they all have inside a semi-fluid substance called the cytosol. Okay, so that's the liquid inside cells. Okay, that's what allows all those chemical reactions that keep a cell alive to happen. It's mostly water, really. Okay, all cells, doesn't matter if it's prokaryotic or eukaryotic, have chromosomes. Okay, so chromosomes carry genes, in other words, DNA. Okay, so that's a very universal molecule here, and they all have ribosomes, and we haven't talked about those yet. But ribosomes are what turn DNA into proteins ultimately. Okay, so every cell has to make proteins, for structure and for function, for enzymes, all that stuff. So all cells are able to do that. Now, the contrast is prokaryotic cells, as I had mentioned before, they do not have a nucleus. Okay, so even though they have DNA, they don't keep it inside a nucleus. All right, so it's in an unbound region called the nucleoid, and it kind of floats around on the inside. I'll show you a picture on the next slide. Prokaryotic cells also lack membrane-bound organelles. Now, we haven't talked about what those are yet, but we will just know that none of the membrane-bound organelles we're going to mention for eukaryotic cells occur in prokaryotic cells, right? So if you look at this picture here, what you're looking at is an example of a prokaryotic cell. So this is your, typical bacteria, and it's a bacillus, which means it's a rod-shaped bacteria. Kind of looks like a Tylenol or something, So anyway, so this is the bacteria. Okay, it's got ribosomes in it, so that is one thing that all cells have in common. It's got a plasma membrane, or what we also call a cell membrane, and it's got a chromosome, which is the DNA in it, right? The other stuff may or may not be shared with eukaryotic cells. Okay, so pili, these are basically how bacteria stick to certain things. Like, if you haven't brushed your teeth in a while, which please do, but if you haven't brushed your teeth in a while, that little film you get on the front of your teeth, it's bacteria that are growing and sticking to the front of your teeth, right? So what do? You put some abrasive on a brush and you brush them off, and that's what, brushing your teeth is, okay? The nucleoid is that general area where the DNA is, but again, it kind of flows within the cell, so I'm not really going to mark that off. Um, some bacteria have cell walls, some don't. There are animal cells that, well, sorry, by definition, do not have cell walls. Okay, but this is like an added layer of protection. This, for bacteria anyway, sometimes makes it really hard for antibiotics to get into the bacteria and kill them. So cell walls can be protective, in other words. Then that goes along with the capsule. Flagella, as you can see right here, these little sort of like whip-like structures that stick out of the back end, these are more for locomotion. Okay, so if you have this cell that's swimming around, it has obviously a lot more benefits than if it just has to stay still all the time, right? The only cells that have flagella that are animal, anyway, or human would be sperm cells. So this is very much like the sperm tail. I think everybody's familiar with that model, where it just kind of like waves in the liquid and just basically swims forward. Same kind of thing for bacteria. So there's a little motor at the base here, which we'll talk about. That basically moves the flagella back and forth and allows it to move. Okay, eukaryotic cells. We're going to spend a ton more time on eukaryotic cells. By the way, if you want to learn more about prokaryotic cells, there's also a microbiology class that you should take, and a lot of you probably will have to take that as a requirement. That gets way deeper into bacteria and viruses and all the little microorganisms that we're really not going to spend a ton of time on in this unit. Okay, so eukaryotic cells have DNA in a nucleus that is bounded by a membranous nuclear envelope. Okay, so in other words, they do have a true nucleus, is really what we're saying here, and that's where the DNA is kept. Eukaryotic cells have membrane-bound organelles, which we'll talk about, as we go forward, as I said before, and they're usually much bigger than a prokaryotic cell. Okay, so in other words, your cells are way bigger, maybe about 100 times bigger or so than prokaryotic cells. So one of the things that I do in class, when we are in class, is I get everybody to look at their cheek cells. Now, you may have done this before, you may not have, but generally a person's cheek cells, so kind of like a little bit regular, I guess. The edges aren't regular, and they got a nucleus on the inside, right? And then a lot of times my students will be like, well, what are all these little dots on the outside, right? Well, like I said, your mouth, like your teeth, are full of or is full of bacteria. All these little tiny dots would be the bacteria that adhere to the cell surface. So this is about how big bacteria are, these little dots, as compared to the whole eukaryotic cell that makes up the inside of your cheek, right? So it just shows you, some size there. The logistics of carrying out cell metabolism sets limits on the size of cells. What I'm really telling you there, okay, or what the slide here is telling you, is that cells have a size limit. They can't get too big, because if they do, they're not going to be able to have enough surface area to let things in to fuel all the processes on the inside. Okay, so you may know this, you may not. The bigger an object gets, the smaller the surface area is compared to its volume. Okay, so we're not going to get down into the calculations of that or anything, but suffice to say, cells can't get too big, otherwise they can't absorb enough from the environment to stay alive. I think that's probably the easiest way to say it. Okay, so the plasma membrane, that membrane that I'm pointing to up here that I just drew the picture of, is a selective barrier. We're going to spend a lot of time talking about the membrane that allows sufficient passage of oxygen, nutrients, and waste, right? So oxygen and nutrients are going to be going into the cell. Waste is obviously going to be going out of the cell, and that will keep the cell alive. The whole volume, the interior volume of the cell, will be taken care of through the plasma membrane. So that's what I was mentioning before. If a cell gets too big, it just won't have enough membrane compared to volume in order to keep it alive. Okay? And then we'll talk about the general structure of this cell membrane. You know, they're calling it a biological membrane here, but we can just refer to it as a cell membrane. It's basically a double layer of phospholipids. And we did talk about this, back in, I guess it was the last chapter, where phospholipids can form a double layer, or what we call a bilayer, right? So right here, I know I'm going to bracket over bracket here, but right here is that bilayer made of phospholipids. So you can see that, it's basically split down the middle here, one layer on the top, one layer on the bottom. Now, what this means for a cell is one layer, the outer layer, or is facing the cellular environment. So the water or the fluid or whatever that the cell is in. The other layer is facing the interior environment of the cell where all the living processes happen. Okay, so that's the difference. Now, do want to make sure you understand that cell membranes are not just phospholipids, right? They also have these little purple, I don't know what they look like, kidney beans or something like that in here too, right? And these are proteins. Proteins are everywhere in the cell, as we mentioned before. And they also have little carbohydrate side chains. That's these little green things that are sticking out. This does, well, multiple functions for a cell. And, we'll be talking about that when we do the cell membrane. And, just to give you an idea how thin this is, if you're looking at a micrograph, this is an electron micrograph of a cell. This is the interior. So just imagine, I mean, this thing is, so small, but you're looking at it, it fills up the whole bottom of the picture. That's how thin the membrane is, right? So there's that bilayer. You can see that little double dark line. And that's what's allowing the cell to bring things in and out of it and stay alive. Okay, so as we go forward, and this is just going to introduce the next little chunk of presentation here. A eukaryotic cell, remember, not prokaryotic. We're not talking about bacteria anymore. We're actually done those. I think we spent like 2 or 3 slides on them. A eukaryotic cell has internal membranes that partition the cell into organelles. Okay, so how you should think about this, if this is new to you, I always try to break down the word. ELLE, is the Latin term that basically means, it's put on as a modifier, and it basically means tiny version of. Okay. Now, obviously, organs, if you look at it over on that root word on the left here, organs are, specialized structures that have pretty much a set job, right? So, like, your stomach is there to, break up the food that you've eaten and digest it, right? Your lungs are there to absorb oxygen from the air that you breathe. Well, cells have structures in them that have individual jobs, right? They're just really tiny. So that's why we call them organelles. So you can almost think of like, a cell, just kind of like a whole organism with all the little organs inside. They're just microscopic, of course, right? So that's why we're calling them organelles. Both plant and animal cells have most of the same organelles with some differences, and that's what we'll start the next presentation with. Okay, so on to number 3.